Okay, welcome everybody to another one of our Thursday Research Horizons seminars from the Department of Plant Sciences. Today we have two PhD proposal presentations. The first one is Ximena Flores. Ximena is a Colombian biologist from the Nueva Granada University. And through her professional development, she's worked on various research projects related to phytopathology and molecular biology. But for the last 3 years, she was conducting experiments with fungal foliar endophytes in cacao and barcoding fig wasps at the Smithsonian Tropical Research Institute in Panama. And in 2018, she joined Dr. Suha Javaji's laboratory to conduct her Master's of Science under the Neotropical Environmental option. This is apologies. We have this is a master's final seminar rather than a PhD proposal. So with that, I give you the floor, Ximena. Go ahead.
Good morning, everyone. Thank you, Professor Valerio, for the short introduction. I want to start this seminar talking about a little bit about my academic experience. I did my master's under the McGill-Smithsonian Neotropical Environmental option. This program supports students that want to do their research on topics related to environmental problems facing in Latin America today. I developed my research on cocoa harvested in Panama. Also, as a requirement of my scholarship program, I did 2 internships, one at the Colegio de Postgraduados in Tabasco, Mexico, isolating endophytic bacteria associated with diseased fruits. And my second experience, I did it at the International Biodiversity Agency in Lima, Peru. I was part of the characterization of mycorrhiza linked with cocoa roots in and their potential role in the cadmium bioaccumulation. Now, getting into my research thesis, I hope that the next time that you enjoy a chocolate bar, you can remember that manufacturers spent around 300 years in developing the chocolate bars that we enjoy today. Due to the great success that cacao has cacao cultivation has become a really attractive business because the rate of consumption has been increasing per year 2.5%. Just Latin American countries contribute to the global production with a 16%, generating to employ to 1 million people, generating a profit of $100 million to chocolate manufacturers. However, the quality of these products is threatened by objective factors like heavy metals, because the tree is taking this cadmium available in the soil and allocating inside of the seeds. In addition, there are some biotic factors that affect the production of this crop. Among them, a main disease called frosty cold rot caused by the fungi Moniloptera already. This disease can cause 30% of losses, but in some circumstances can reach 100%. This disease affects only the fruits, and this is critical because inside of the fruits are located the seeds, and the seeds are used as basic raw material to prepare the chocolate. So the susceptible stages for this disease are the immature stages from the fruits that are A and B. The spores germinate and cross the layers, the thin layers of the small fruits, infecting the internal layers, causing the formation of the fruits, later internal and external necrosis. Cultural and chemical control haven't been enough to eradicate this disease, reason why scientists have been exploring the potential of endophytic microorganisms. Several fungi strains isolated from cocoa leaves have demonstrated to be successful to control Phytophthora, but just for a short period of time. Another scientists have been isolating strains like Bacillus cereus from tomato and potato, and they reduce the disease just in lab conditions because these bacteria weren't isolated from tropical countries or tropical crops, where cocoa is grown. So this bacteria has not adapted to the weather conditions where cocoa is harvested. For this reason, we decided to explore the biochemical and antagonistic potential of bacteria from cacao fruits. In order to accomplish these goals, I started doing the isolation and biochemical characterization to ferment and dry cocoa seeds. I came to a commercial brand called Jupic, located here in Montreal, to get fermented and dry organic cocoa seeds from Peru. These seeds belong to the variety CCN51. This is an Ecuadorian variety. And the reports mentioned that records high productivity per hectare and is moderate resistant to diseases such as witch's brown and frosted pole rot. With these seeds obtained in the Canadian market, it was possible to standardize protocols of disinfection and method of fermentation, and to have an idea about the abundance and diversity of bacteria that can be found. All the isolates that I got were partially sequenced in order to identify them. And as a result, I got 53 isolates, which has been reported to be part of the microbiome community, even under other species like coffee, tomato, and tobacco. Most of the strains isolated belong to the phylum Firmicutes, which includes Bacillus, Enterococcus, Pinobacillus, and Staphylococcus. This group of microorganisms has been reported to be growth promoters, or some of them express antagonism role against some pathogens. Another main group were Klebsiella and Enterobacter. They are also reported to be part of the rhizosphere in another plant species. And when they are available in the soil, they can fix nitrogen. So as an interesting point, I want to highlight that I got 12 different species of Bacillus. So these Bacillus has been reported lately to be part of the organic fertilizer, because these Bacillus are able to produce spores which are resistant to desiccation. Moreover, most of them are growth promoters, and in some circumstances, some of them can reduce some main diseases. So in order to confirm this research based on bibliographic reports, I ran some biochemical tests as a part of my second objective. I evaluate different tests designed to categorize the microorganisms based on their biochemical response. Inside of the plant colonization traits, I evaluate the ability of releasing lytic enzymes. Inside of the growth promotion traits, the ability to release phytohormones and getting micronutrients from the soil. And inside of the antimicrobial traits, I assess the ability of the bacteria to release toxic compounds and and other small molecules like siderophores that spread the ability to compete in the environment. I wasn't able to do the antagonism assay because we were waiting for the export permit that still was pending, and the pandemic times arrived, so the COVID delayed the whole process. As a result, here you can observe the production of enzymes and metabolites involved in the different traits evaluated. One single strain can be successful in more than one test. Now we are going to see the proportion of the positive bacteria per test. After collecting whole data, we can discuss several aspects. The ability to produce enzymes like cellulose, amylase, and protease is a trait that gives to the bacteria a great adaptivity because they can list the cell walls of pathogens. In addition, the secretion of alginate and cellulose are involved in the biofilm formation. Now, regarding to the antimicrobial test, as you can see, a great proportion, 21 of 53 microbes are resistant to ampicillin. And this antibiotic is the broad spectrum and these interfere with the cell wall formation. Moreover, most of the strains, not most of the strains, but a great proportion, I guess more than the half, are able to produce chitinase. So it means that they can lease the cell wall from the fungi and some insects. The siderophore production is an interesting trait because it has the ability to sequester ions from the soil. And this micronutrient is difficult to take from the plant. Plus, can take it from the soil, making a competition against the fungi that always need it. And a low proportion of bacteria are able to produce AGN. This is volatile and that inhibit strongly the cytochrome C. Only 7 strains are able to produce, but based on bibliography reports, this trait is commonly found in rhizosphere bacteria. Now, talking about the plant growth motion test, the enzyme ACC deaminase ensure that the ethylene levels doesn't come too elevated, helping to the formation of longer roots, in this way enhancing the survival of the seedlings. All of us, we are familiar with the role of the phytohormone indolacetic acid. But in this special case, I want to highlight that bacteria that are able to produce more than 100 micrograms are potential to be or to help in the plant in the bioremediation process because they can take cadmium or heavy metals present in the soil. So followed by the calcium stabilization. This is an important trait now, even now, because we are facing weather conditions changing all the time. And the bacteria are able to sequester the calcium, making to the water available to the plant. And finally, the organic acids is also important because they can degrade some nutrients that are not available to the plant, making reducing in the way that are available to the plant. So to sum up, I select a group of microorganisms with exceptional biochemical traits based on the indolacetic production and siderophore production, because these group of microorganisms in the future can be a potential fertilizer for the farmers because they exhibit traits that help the growth promotion. So with this information in our minds, I decided to go to Panama in order to explore the bacteria associated to the layers that are shaping the fruits. So from Panama City, I traveled with Dr. Luis Mejia, who is the leader of the project Cocoa Microbiome in Panama. We traveled to Bocas del Toro because there is located the farm, the Magnita farm. Mr. Lozada is the owner, and he has several varieties of cocoa from countries like Costa Rica, Colombia, Ecuador, and Panama. So taking advantage of the variety of cultivars that Mr. Lozada has in his farm, we decided to select 2 varieties based on their response against the pathogen. We select KTR6, that is a tolerant variety, and Katongo. This variety was originated in Costa Rica, and Katongo is mostly grown in Brazil, and this variety is susceptible to the disease. We select 10 fruits, healthy and mature, in order to explore the internal layers. So I explored the endocarp, the pulp that is covering the seeds, and the seeds. Once we reached the lab, I did the store-first sterilization of the fruits, and I opened them under sterile conditions. I extract small pieces of tissue from the endocarp from opposite sections. I took the pulp from 10 seeds and 10 seeds. I followed the same protocol in order to get the bacteria. So I homogenized the tissue, and later I did serial dilutions, and I played the dilutions in 4 different media in order to capture the highest diversity of bacteria present in each layer. All the isolates were molecularly identified. And as a result, we can see that most of them, of the microbes are associated with the seeds. I got 160, but only 91 of them were partially sequenced, or I was able to get information from them. So most of them are located in the seeds, followed by the pulp and the endocarp. It's interesting to know that there is no big difference in the abundance of microbes between the two varieties. So here, I want to show you the variety and the proportion of the microbes in each layer. So most of the microbes, they belong to the phylum Pharmacodes, followed by the Proteobacteria. So followed by the bibliographic reports, most of them can play a potential role as a growth promoters. But later on, I'm going to show you the difference between them. Here is the same situation with the susceptible variety. Most of them are Pharmacodes followed by Proteobacteria. And they are the common core microorganisms that you can find in other species. And they play a role as a growth promoters. Here is the important part. Here's the message to take to home. So first of all, the bacteria colonization is driven by host genotype. So in the first line, I put in the bacteria that are specific isolated in the tolerant variety. In this case, I want to especially highlight the presence of Coccasonia, because in one report, this bacteria was isolated in tolerant varieties of papaya, and this variety was tolerant amine disease caused by Erwinia. Another important bacteria here, interesting one, is Acinetobacter, and look, is the presence are in the 3 different layers. And these bacteria, in vitro conditions, scientists has collected some metabolites that play a role as antagonism against fungi like Fusarium and Botrytis. In the middle, we can see the common bacteria between the 2 varieties per layer. So these bacteria, Pseudomonas, Cluberia, Klebsiella, Enterobacter, and Pantoea, they are part of the common core of the microbes associated as endophytes in different crops. I want here to highlight the presence of Terribacillus and Rosenbalia because they are specific from cocoa. Until now, there are just 1 or 2 reports of this group of bacteria because they are involved in the fermentation process. They are the starter of the process. And in the last line, I'm showing you the bacteria that are only present in the susceptible variety. It's kind of intriguing that in the seeds of this variety, there are a great diversity of bacteria. So reading and looking for some explanation about why this can be happening. So I found that some researchers in Brazil doing the characterization of the, of the seeds in terms of the chemical properties. And they found that the seeds of this variety have a low proportion of theobromine and phenolic acids, So it means that this character make them a little more susceptible to be colonized by other microbes. That could be the main reason why we can see a great proportion of different genera in the seeds. So we weren't able to perform biochemical tests because the spore permit was pending. And again, COVID time delayed the whole process. So I did a research in silico to make a prediction about the potential role of these bacteria in the plant, in association with the plant. And most of them can play a potential role as a growth promoters, followed by colonization traits. I want to show you that my results are the first a study on the microbiome unsusceptible and tolerant of commercial varieties grown in Panama. This is the first study that shows the different the bacterial composition on tolerant and susceptible varieties, and this information can be valuable in the breeding process because bacteria are driven by host genotype. And this is the first study on microbes associated with tissue-specific in bacteria, because there are several reports, but they are associated with the seeds or the boxes of the fermentation. As a future perspective, I would like to encourage people that are doing research on cocoa to perform antagonism assays using the strains that were isolated from the internal layers of the cocoa. Plus, could be really interesting to have the opportunity to test the ability of the Klebsiella, that was the general bacteria that has the ability to release a high proportion of indole acetic acid, and just to know if these bacteria can remove cadmium from the soil. What else? Make a comparison of the metagenomic analysis from the different tissues, and just to know what important and valuable information we are missing, because there are no cultural microorganisms, and test consortiums of my top 6 microorganisms that I show you to provide a potential organic fertilizer to cocoa farmers. I want to express all my gratitude to my supervisor, Professor Suhaya Baji, for all her support during my experiments and my travels, to my co-supervisor, Dr. Alan Herre and Dr. Luis Mejia, for allowing me to be part of the project Cocoa Microbiome in Panama, to my committee member, Dr. Valerie Gravel, to all members from the Fund Lab, previous and current ones, and the nice and really helpful team from Smithsonian and INDICASAT, Mr. Lozada, for allowing me to collect my samples there. And a special thank you for Professor Sharon and Erin Minet for all their support during the time that I was facing in Lima, the COVID time, and all of my agency and funding members. So I will be happy to receive some questions.
All right, thank you, Ximena. Okay, at this point, you can write any questions into the chat. If you're on YouTube, you can also write it into the chat and I will pass it on. Or you may also unmute yourselves and ask a question.
Professor de Blois here.
Yes.
So thank you very much, Ximena, for your presentation, and congratulations for having been able to complete that research in challenging conditions. I was curious about, I'm not an expert on bacteria, but in your last project, you compared 2 varieties, right, that were grown in the same, at the same location?
Yes.
So my question is, if these varieties, for instance, had been compared between different locations, had been grown at different locations, would you have expected similar patterns? Because I think we found many things in common, right?
That is a pretty interesting question. But definitely, the soil conditions make a difference in, in the colonization of the plants. But for instance, when I was checking some reports available in Africa about the fermentation process, but because it's the only information that I have about bacteria, they show a strong difference between the farms in the different varieties. So definitely, I will find different colonization in different places. But also, it's important to mention that the colonization of the varieties is driven by host genotype. So there always be a core that is common per each variety. And we decided to run these tests, to run these experiments, just taking advantage of the varieties that are present in the same place, because it's difficult to compare, to compare varieties in different places. Just we want to know how the microbiome are associated just in one place. Because we can control all the variables, And we want to know what, in this case, what factors are driving the host colonization.
It is like a common garden experiment.
Yes.
Thank you.
Any other questions for Ximena?
I think I have a question. So it's based a little bit on Sylvie's question. So you mentioned that you saw differences between the two varieties, the two cultivars. So one that is susceptible and one that's not susceptible. Do you think there are other factors other than susceptibility and tolerance that could also explain the differences between what you found?
Yes. more than the character of the resistance or the susceptibility of the tree, I consider that the chemical composition of the fruits play an important role, like the proportion of the theobromine, proportion of the phenolic compounds, can shape the chemical of the tree can shape the microbes that are able to colonize the tree.
Okay, thanks. Maybe another question if no one else wants to ask a question. You also mentioned the fact that, in your future work, what would be interesting is to maybe use a technique that could look at the microbes that are not culturable. Mm-hmm. So do you have an idea of the proportion of microbes that you were not able to look at that would be microbes that are not culturable?
Yeah, definitely. But thank you for this question. Recently, there are a report just focused on the transition of the microbes during the fermentation process. And this is very interesting because the composition of microbes change along the time. So they start, for instance, in the first 48 hours, they just found bacteria and yeast. And after 44 hours, they start finding only lactic acid bacteria. And after that period, acid bacteria. And at the end, all the communities decline because the temperature is reached around 50 degrees. So this part is interesting and fascinating because all the microbes are not constant. And I just evaluate the first the endophytic associated with the layers without any chemical or without any intervention process. But that definitely, I bet that there are another microbes that need special conditions to appear in the environment.
I'm just curious. Is there? I believe there is a French group to sequence the cacao genome. Did they find anything in terms of resistance genes that are also associated with resistance to Phytophthora, for example, in other species like potato, for example? Like, in potato, there's a lot of effector genes that were detected after the genome sequence, which are associated with gene for gene type resistance.
Okay. Well, there are more than one gene. So here, as I mentioned before, the fungi faces a biotrophic and a neutrophic phase. So during this time of period, the fungi can be inside of the fruit without causing like any necrosis that you can see. So one difference between the tolerant and the susceptible variety is in this shift between becoming biotrophic and neutrophic phase. Why? Because there are some genes in the plant, the chitinase. So when the plant starts or recognize that it's infected. So it start releasing hypersensitive response, carotenase, and all the chemical compounds to fight against the fungi, right? And this response is triggered. So in the case of the tolerant variety, the response is not too strong. And at some point, the response from the plant is not too devastated. Because when the plant start fighting, so the fungi become really aggressive and start forming the internal and later the external necrosis. So there are several genes, of course, implying the response. Indeed, there are Penn State is working on it, and they are trying to do CRISPR on specifically with the penetration genes. That is like
Okay.
the first barrier to cross the pathogen.
Okay, that's very interesting. If there are no other burning questions, and I believe there are not on YouTube, then we will we'll move on to the next seminar. So Marianne, go ahead and share your screen.
All right, let me just get this organized. Share my screen. Here we are. And now, this.
All right. So Marianne will be presenting her master's final seminar titled Monarch Butterfly Distribution and Reproduction in Eastern Ontario. A little bit about Marianne. She obtained her bachelor's in ag environment and environmental biology at McGill. She's completing her master's in Dr. Sylvie DeBlois Landscape and Plant Ecology Lab. She's co-advised by the director of the Montreal Insectarium, Dr. Maxime Larivet. And Marianne worked also as a science journalist and educator, specializing in nature and environmental issues. Okay, go ahead, Marianne.
All right, can I ask you what you are seeing on the screen? I'm trying to get the presenter view up, but
yeah, you need to put the presentation view.
and of course, having worked many times in the past, it's now not working. I'm wondering what I did wrong. All right. Give me just a moment to try and work this out. No.
Press F5.
Yep, I'm doing that and it's not working. Okay. Isn't that frustrating? What am I doing wrong? Let me see. I'm just gonna shut it down. All right. And try and reopen it. Give me a minute. Of course, this happens. There we go. All right, let's get this up. Aha. And now, if I go, oh, hang on, I need to figure out Alt-Tab. I should be able to share my screen. Aren't we having fun? Share screen. My presentation. And it's back in the other one. Let me try again. Worked beautifully when we tried it before. All right. I'm just going to have to go with the slide. Let me try the slideshow. If I do Alt F5 now, you can, now you're seeing my,
yes,
now you're seeing my screen?
Yes. Perfect.
Okay, off we go. Excuse me if I stumble. All right, welcome. Thank you for listening. I'd like to share my research into monarch butterfly distribution and reproduction in Eastern Ontario. So Danaeus plexippus is a iconic butterfly known around the world with many unique features, including the fact that the female butterflies ovipost only on Asclepias, members of the Asclepias family. So those are the milkweeds, which is the food source for the caterpillars. The other fascinating aspect about this species is that monarch butterflies born here migrate all the way down to a concentrated area in central Mexico, where they overwinter in small areas before beginning the migration back up north. They lay their oviposts, lay their eggs in about southern US, die, and then succeeding generations move north before arriving in our study area. And if I can show you, I hope you can see my cursor in this situation, but the group that I studied would come north of Lake Erie here to the North Shore of Lake Ontario. Now, the interesting thing about that overwintering site is, unlike many varieties of insect where you cannot get a sense of the total population, here you can measure each year and compare what area they're occupying. Unfortunately, the results demonstrate that the monarch has experienced greater than 85% decline since we started measuring. We know they're in trouble, but we knew very little about the situation here in Canada. So that's where my research came in. The initial question I was trying to answer was, what are some of the factors explaining the spatial distribution and reproductive preferences of the monarch butterfly in Eastern Canada? And I was looking at this at both the regional level using community science data and then field sampling. So for the regional level, we used a community science data set of observations uploaded to online websites such as eButterfly and the Montreal Insectarium's Mission Monarch, using recent observations. And we used both observations of monarchs, but then observations of butterflies other than monarchs in order to get a presence and absence data set. And some of the initial research done by the Insectarium showed that there was a hot spot on the northeastern part of the North Shore of Lake Ontario. So I took my data set, limited it by the extent of Lake Ontario, which resulted in about 1,800 observations. So the research questions at this level are, is monarch occurrence related to land cover at this scale? And can I recognize that monarch hotspot again? And if so, can I relate that hotspot to land cover? So for land cover data, we used the map from the Commission for Environmental Cooperation. This is a 2015 map of about 30-meter scale using ArcGIS, and we drew a 30-meter radius buffer around the observation and then extracted the land covers found in that area, which resulted in about 900 sites. For my data analysis, I used the ANOVA, but also logistic and generalized linear regression. And use multimodal inference to compare the regressions. This is known as model selection. So what you can do is use Akeiki's information criterion to value different models. So you can run regressions, but how do you compare them? So this weighs models with variables that describe more of the variation. It also upweights or gives greater weight to models that are the simplest, use the fewest variables to describe. So you can also compare groups of models. So we created several groups of models representing different combinations of habitats. So forest habitat, open habitat, crop, urban, and water we analyzed separately. And by urban, I'm referring to both built-up areas and as roads and trails, anything human influenced. I did a separate analysis of geographic coordinates and distance to water. So is monarch occurrence related to land cover at the regional level? We expect that occurrence would be increase in open habitats. So literature says that the monarch is a creature of open meadows. If you had giant sails on your back, you probably wouldn't be too fond of forest either. And so we would expect that they would decrease in forest. And this proved to be the case. So monarch occurrence, your chances of seeing a monarch decrease significantly in needle forest and deciduous forest, and not significantly in mixed forest. So they also decreased in urban areas, and they increased in crop and the open habitats, but not significantly. It didn't describe a lot of the variation. In the spatial analysis, monarch occurrence, you were less likely to see a monarch the further you got from water, and the further north you went and the further west you went, which implies that they're not evenly distributed in space. So that led us to look for the hotspot. So we used hotspot analysis, which identifies concentrations in space of occurrence. And it also recognizes cold spots, so areas where the opposite is true. And if you look, I can show you, I hope my cursor is working. So these red blotches here are the hotspots, observations in a hotspot. The little freckles are just neutral points that don't have no values. And down here in the bottom, I hope you can see there is a blue, and up here there are blue areas that represent cold spots. So if I bring up the land cover so you can orient yourself a bit, you're looking at Lake Ontario. The hotspot, for those of you who know Ontario, would go from Prince Edward County through Peterborough up to the Kawarthas. This red smudge here is Toronto, and then the cold spot is southwest of Toronto heading towards Hamilton. So could land cover tell us about anything about the probability of being in a hotspot? So we would expect that if the open habitats would increase the probability of being in a hotspot, and the forest would decrease it. And this, the probability of being in a hotspot decreased with being in the needle forest and the deciduous forest, though not significantly, but it increased significantly in mixed forest and cropland covers. So I did an ANOVA to compare the land covers in the hot and the cold spot. And it wasn't significant between the regions, but within the regions, there were significant differences. So if you look at, this is a log measurement, and this is, the yellow is deciduous forest and the brown is urban value. So you see that they are significantly greater than all the other ones. So the open habitats are just negligible in terms of overall land cover. And the only interesting thing is that in the hot spot, you have considerably more, a significantly greater amount of crop. So hold that in your head as we go on. We decided we wanted to look at this on the ground. So we did field sampling and comprised the cold spots, the hot spot, as well as some of those points and neutral points in the middle. And we were also interested in the difference between urban gardens and rural areas, because monarch or pollinator gardens have been very popular. So we took a set of points in Toronto as well. And at this level, I wanted to know, is juvenile and milkweed abundance related to region or land use? Is juvenile abundance related to plant species or habitat characteristics? And to do the field sampling, we used the integrated monarch monitoring protocol of the Monarch joint venture. We looked at 82 locations. So these were randomly chosen but binned to ensure that within each region we had sites with very few monarchs and the highest number. We went twice, so in June, July, and August, and gathered information on 64 possible variables. So encompassing environment, land cover, land use. So we made a differentiation between rural land areas, urban parks, and gardens. The structure of the site, so what percentage of grass, forbs, shrubs, trees, wetland it was. We walked a transect and counted adult monarchs, and then turned over an enormous number of milkweeds to count juvenile monarchs. We also analyzed the plants using 12 meter square plots, 4 in the center, 4 in the middle, and 4 in the edge, or counted the plants rather, and identified them. And so my first conclusion is that monarch butterflies are found with thistles, poison ivy, and wasps. It was, I can confidently say that the poison ivy resources in Eastern Ontario are not in any danger. We also immediately discovered that the open habitats, that regardless of what the land cover, the remote sensing land cover was saying, that these sites were in open habitats. We also discovered that counting adult monarchs is a difficult process, and they are very particular. They don't fly if it's too hot or too cold, too late in the day, and so we would need to go back more than just twice in order to assess the population with any confidence. So we chose as our response variables the juveniles, because they are sort of imprisoned by the milkweed. And so our first question was, can those regions describe any difference in the juvenile monarch population? So the answer is yes, sort of. If I look at juvenile monarch abundance by region, so just a count, and most of the site areas were pretty comparable across the different regions. There is a significant difference between the number of the abundance of juvenile monarchs in the hotspot and the midpoints. But I add a measure of density. So I would look at juvenile monarch density on the milkweed. So how many monarchs were found on each milkweed stem? Then Toronto has considerably more denser population than the cold spot. So this would seem to indicate that, well, first of all, the literature says that Zaluki and Kitching say that a female monarch will preferentially ovipost on isolated milkweeds. So the implication is that if they're having to ovipost on more than one egg on a milkweed stem, that the pollinator gardens in urban areas could be vital because the resource is limited there compared to rural areas. And so I decided I wanted to have a better look at those gardens compared to the rural areas because of this possibility of resource limitation. And if you look at my picture here, these are two late instar caterpillars that are just taking this milkweed back to the stem. And I wanted to give you some examples of what we were thinking about the difference between land use. So this is my favorite outlier in urban Toronto. It was this ancient couple who had 3 stems of milkweed in their backyard, but there was a dozen juveniles on it. Compared to milkweed found in open areas where you see the milkweed actually likes to social distance. There is about 6 feet apart normally. So when we decided to look at milkweed, how milkweed varies with land use. And we found that milkweed, once again, it followed the region. Abundance was significantly greater in the rural areas compared to the gardens, but milkweed density, so milkweed stems in the 12 meters squared plots that we took, was significantly greater in the gardens compared to the rural areas. So this would seem to point out the value of both rural areas where you have more juveniles on more milkweed, but also the importance in urban areas where you have more juveniles on a single stem, and those stems are concentrated in space. So the need for pollinator gardens, especially in a place like Toronto, which appears to be kind of on the flyway where they're migrating north, seems to be vital. So I wanted to also consider what other plant characteristics might affect juvenile abundance. So could juvenile abundance, how did juvenile abundance change with plant and nectaring plant richness? So we would think that with both, their abundance would increase, but in fact, it was not the case. In the case of juvenile abundance increased significantly with plant species richness, but it decreased significantly with nectaring plant species richness, which was a surprise. The implication is that the adults who use the nectaring plants have slightly different requirements from the juveniles. And if we looked at how that distribution occurred in land use, you'll see that rural areas had a significantly greater species richness than garden. So we decided to have a look at plant species composition more closely. So I gathered a set of nectaring plant species that were common and abundant in my data set. and analyzed those, expecting to find the juvenile abundance increased with increasing nectaring species of open habitats. Well, it was not the case. Juvenile abundance decreased significantly, often with most of the species of the nectaring species of open habitats, except for rubus species, raspberries, and Prunus pennsylvanica, so pin cherry. And those are not so much found in open habitats, but edges. They need sunshine, but some humidity. And so I thought, well, maybe edges are an issue. So I gathered a set of, made a second model out of species that were common and abundant in my data. And sure enough, the juvenile abundance decreased significantly with the nectaring species and increased significantly with species found on the edges. So the why, the implication is that the females are preferentially oviposting on the edges, or alternatively that the juveniles on the edges are experiencing greater success. They're just more successful there, and we don't see them as much in the open habitat, drier open habitats. There's not a lot of literature on this. There's some research in India demonstrating that certainly caterpillars are related, caterpillar success is related to humidity. And there's a study by Potter that shows that, Baker and Potter, pardon me, that shows that females will preferentially ovipost on milkweed at the edges and prefer that to like a tangled landscape. I didn't look at that, but I thought, well, is this saying something about habitats? And can I find some interesting effects in environmental attributes and juvenile abundance? So the hypothesis was that juvenile abundance would increase with increasing shade in trees and decrease with forbs and distance to water. And that proved to be the case. The further juvenile abundance increased with proximity to water, with the percent shade and with the percent of trees, and decreased with the percent of forbs. And as a real life example, I show you these two photos. So this is a site in the hotspot. The road is oriented north to south, and there's milkweed on both sides. But one side only got early morning sunshine, whereas the other side was in the sun much more of the day. And there were many juveniles, but only on the shady side. So in conclusion, rural habitats offer more milkweed, but urban sites tend to concentrate plant resources. The urban sites have more juveniles per host plant, which may increase competition and emphasize the importance of those urban sites. Juveniles and monarchs may have different habitat preferences, so they all use milkweed, but in different places. Juveniles may prefer shade or open edge to shade or edges to open habitat, or they may do better there. So in Eastern Ontario, the best sites for monarch observation and reproduction would be botanically rich open habitats near water, offering edges with some tree cover. So I suggest you go out there and upload some observations like these people did. So one of the great joys of doing the field work was I got to meet some of the people who uploaded their observations to the websites, and that was great. But I would also like to thank my advisory committee, as well as my funding bodies, the Insectarium, the Fonds de Richesse de Québec, and NSERC. I would also like to thank my daughter, Mae Boley, who was my assistant milkweed counter on some of the sites, as well as the many people who have been involved in given their time and their knowledge to assist me in things like GIS and statistics analysis, and as well as the plant identification. Thank you.
All right, thank you, Marianne. Again, if you have any questions, please, you may unmute yourselves or write them on the chat. For those on YouTube, again, write the question on the chat and I will pass it on.
Hi, Marian. This is Vinko.
Hey, Vika.
Hello. Great presentation. Really enjoyed it, and definitely learned a few things I hadn't realized about monarchs. So
Yeah,
thanks for that.
my pleasure.
I'm curious about your observation and what you found in the literature that some monarchs, adult monarchs, tend to prefer ovipositing on solitary milkweed plants, and we talked a little bit about how that can increase, how that relates to competition, and so I'm curious what your thoughts are about the trade-offs between the interspecific and intraspecific competition, or sorry, not inter and intraspecific, but the competition between the offspring of a single individual and the competition between the offspring of multiple individuals. You know, I would first think, hey, I'm a monarch. I want to go oviposit wherever there are a lot of milkweeds. But do you think that that competition between adult individuals is so great that that's the reason there could be a preference for individual plants? If that made any sense.
Well, if you watch them oviposting in a field, for example, it's a delight, and we would chase after them trying to find them because they will only usually ovipost one egg per milkweed. So you see them, they land, they lay an egg, move on to the next one, move on to the next one, move on to the next one, and they do a transect across a field. And part of the reason why this research is known is because it was discovered in the Midwest that they preferred to ovipost on, you know, you would have a whole tangle of milkweed in the road edge, and then you would have, before modern agricultural methods, you would have, because milkweed was formerly known as, you would have stems that would come out into the fields, and you would inevitably find more eggs. Those isolated milkweed would have more eggs than the ones that were in a tangle. And we found that as well. You know, you would find this glorious bunch of milkweed, and you would expect to find them there, and then there would be some poor tiny little milkweed that had been mowed, and that would be where the eggs would be. So, and we also would find you would find milkweed that was just a stem, you know, so competition or predation. I didn't look at predation, and parasitism may be an issue. But it is thought that that's why they will do that, and it's a noticeable effect. They will ovipost more than one on the same plant, but so if there's already eggs on a plant, a female will ovipost on that plant, but she will only ovipost one egg per plant, and preferably on the ones on the outside. So does that answer your question?
Yes, thank you very much.
Okay, we have time for one more question. Alessandro.
Hello, Marianne, it's Alessandro. How are you
doing?
I have a small question. You kind of answered it with the previous question. You didn't look at predation. Could the habitat affect the predator density, and that would also have an impact on larvae density? Because when you see larvae, it's larvae that survived till the state that you looked at, and not eggs that are laid on the plant, which will be a more precise information about how can I say use of monarchs of the milkweeds that are placed. So do you believe that maybe the habitat could affect predation and then affect the quantity of larvae that you've measured?
Yes, there is so many different ways you could go with this data set that I'm a little overwhelmed, but I did take, I note how many eggs and how many larvae, and I believe it's understood, like there's a understood rate of, you know, if you have X many, I think it goes, if you have a certain number of like 4th, 5th instars, so the larger caterpillars, you can infer from that how many eggs there are, and the reverse, right? So, and I think what you would want to do is measure, you know, check that across the different habitats, different locations, to see how does the rate that we observe compare with the known rate, and that would give you some idea, and then compare, see, okay, are we finding a greater a difference in that variable that is dependent on the types of land cover. That would do it. You could do that with this data set for sure.
Great, thank you.
Okay, well, that takes us to the end of today's seminar sessions. Thanks everybody for listening and attending. And don't forget to fill out the survey on my courses for Marianne and Ximena. See you next week. Next week, thank you, Marianne. And next week, we'll have Dr. Harkam Al-Walia, Associate Professor. He's the Harriman Chair of Agronomy in the Robert B. Doherty Water for Food Institute Fellow in the Department of Agronomy and Horticulture at the University of Nebraska. And so please, if you're interested, join that talk. Should be very interesting. Thank you, everybody.
Thank you. Bye.
Thank you.
Bye.
Valerio, I have an announcement.
Oh, yeah, go ahead, Suha.
Okay. I will be the co-instructor for seminar 691 in winter. And I would like those who are presenting now, the student, you need to register for this course. If you can't register online, you will not be able to see the presentation on my courses, nor to fill in the evaluation survey. Please do that. Discuss with your advisor, because you cannot present a seminar without having a first committee meeting. And I already put all the staff members as research assistant in order to be able to go into my courses. This is a moving target. I tried with Erin to find out who is going to present in winter, but honestly, they don't know, because sometimes the committee meeting has not been signed and she doesn't have this information. So I urge those students who will be presenting in winter to register, and even those who had registered in the fall also register, because now on my courses, I don't see any students. So please do register and discuss with your supervisor when you are going to have a committee meeting. And I will try, once you have the committee meeting, I will try to schedule your seminar 1 to 2 weeks after the committee meeting. And thank you, Valerio, for the good work of the fall session. And you are already a co-host, co-instructor in the winter session. This is what I want to tell you.
Okay, so Valerio, I think he's gone. He had a meeting at 11, so I guess I will be the one ending the session. So have a good day, everyone.
Okay, thank you.
Okay.